Why this happen? Oh, this is DNA cloning. DNA cloning is the process of creating multiple copies of isolated DNA fragments by in vitro or in vivo method. DNA cloning, also known as molecular cloning, gene cloning, recombinant DNA technology. What are the components needed in DNA cloning? They are DNA, vector, description enzyme, and DNA ligase.
Now we are proceed to steps in DNA cloning. The first step, DNA of desired gene is isolated from DNA. Second step, isolated DNA is purified and fragmented with restriction enzyme, producing staggered cuts in specific sequence and generate fragments with cohesive ends. For step 3, each ends of the fragments have capability of hybridizing with fragmented DNA that have the same restriction enzyme. Step 4. DNA fragments are incorporated into plasmids.
So, for step 5, single restriction site for plasmid cleaved by restriction enzyme is then generating the same cohesive end as cloned DNA. For the step 6, enzyme and DNA ligase is used to form phosphodiester bonds. For step 7, the plasmid are incorporated into the bacterial host by transformation process. For the final step, each cell that contains a different segment of DNA from the original organism is taken together, representing the DNA library. It then can be plated out on an agar medium and identified and isolated.
Advantage of DNA cloning. For human, DNA cloning allows scientists to harvest an entirely new functioning organ to people on the organ donor waiting list. Besides, gene cloning also able to create many new, advanced and helpful pharmaceutical products which very important to human. Example of pharmaceutical product is human. For environment, engineered microorganisms can help us in coping with some environmental problems. For agriculture, DNA cloning can produce transgenic plants that resist to drought, frost, and other problems. In economy, DNA cloning can increase the production and quality of the product. Example is improving milk production in cow. Another advantage for DNA cloning is can prevent shortage of food crisis in future.
Let's continue. One of the biggest disadvantages of cloning is technology is still uncertain. Scientists still unsure of genetic mutation that might occur. There were hundreds of failed clones including dead fetus and turned out horribly deformed. Losing gene diversity is another disadvantage of cloning. If gene diversity is lost due to the excessive cloning, there are no natural mutations to allow clone to survive new disease because of low immunity. Then there is always risk of cloning technology being abused. Imagine corrupt dictator could do with cloning. There will always someone looking to use cloning for their own personal use and profit.
Dolly the sheep is the world's most famous clone. It is the first mammal to be cloned from an adult somatic cell using the process of nuclear transfer. Dolly was born on 5th of July 1996 and was euthanized on 14th of February 2003, aged 6.5. How Dolly was cloned? To do the cloning, scientists used an udder cell from a fin dorsate white sheep and reprogrammed the udder cell. After that, they inject the cell into an unfertilized egg cell came from the Scottish black-faced eel which had had its nucleus removed. They made the cells fuse by using electrical impulse. Then, they culture it for 6 to 7 days to make sure the cell developed into an embryo before implanting it into a surrogate mother.
Free created using Powtoon.